Fine. It's really nice to do another one of these. And this is quite a special one for me because it's the first time we've had a speaker who we did our PhDs together a number of years ago. So it's absolutely lovely to have Kostas, who's currently in Switzerland, which is why he's doing this virtually. For those of you briefly who haven't been to one of these seminars before, we do these every couple of months. Really, all they are is just to have hopefully a nice hour out of your working day to have hopefully an interesting talk and donuts. So please do help yourself to the aforementioned donuts. One thing that we're really happy about as well, it's the first time that we had anything concrete come from these lectures. I actually went over to the MRI last month to do a workshop there. And that's the first thing that was actually generated from doing these. And quite honestly, the more we can get of that in the future, the better. But this isn't about me waffling on. I'm going to let Kostas introduce himself. But Kostas, thank you so much for joining us. It's really lovely to see you in a professional capacity again, as opposed to just a catch up. So, hi, are we in the correct meeting? If it's the AI in healthcare meeting, yes is the answer to your question, Kai. Anyway, Kostas, I'm going to hand over to you. Do you want to share your screen? And I'll
yes.
To you.
Yeah, thank you very much. Okay, let me try to share the slide deck. You can tell me if you see it. Okay.
We can do that.
Perfect. Yes, thanks a lot. It is thanks for the invitation, John. It's great to be even remotely. I hope next time we will be in person. Yeah, I did my PhD with Gavin, who is in the audience in the University of Manchester in computer science. And we focus in developing some methods for machine learning. And then I stayed 3 years in Manchester doing a postdoc. And in one of these postdocs, I work also with Matt, who is also in the audience, Matt Sperrin. And in the other, we work with AstraZeneca, and it was in the first time that I started working with clinical trial data. And one of the questions that we focused was around this subgroup discovery, which is also the topic of this presentation. And 6 years ago, I moved to Switzerland. And the last 4 years, I worked for Novartis in the methods group. And a large part of our work is to develop this type of workflows for assessing treatment effect heterogeneity. And we try to apply it in various projects of the company in clinical trials of phase 2 and 3. And I will provide more details. So I will start with a presentation. You know, if you don't hear me or whatever, let me know. So this presentation will split it in 2 parts. The first one will be discussing the workflow that we have put in place, that we use this acronym WATCH for assessing treatment heterogeneity, which is published in Pharmaceutical Statistics. And the second part is one implementation of this workflow that we put in archive earlier this month, and it builds upon some way of inferring the individual treatment effects. So the second part will be more rich in terms of statistics and machine learning. The first part will be more motivating, let's say, more the problem and what we feel that there is a necessity for a workflow. And I will start with some imaginary scenario and to try to motivate the underlying, let's say, need for this workflow. Let's say we have a clinical trial, and we have this disastrous news that the trial failed. And after the initial shock of the team, then try to digest a bit the results, there will be always some person that is very skillful in going deep into the data. And he found that for women over 50, the response rate in our drug is 70%, and while in placebo, it's only 30%. So let's go and give the drug to these people. And of course, as a statistician or data scientist, at least my first reaction, I was feeling very nervous with this. You can feel that something not exactly right going to this direction, but at least the first period that I was engaged in this type of project, I couldn't also clearly specify why I was having this nervousness. So this presentation will be partly also to explore why we have this nervousness and how we can put in place some sort of structured approaches to have more confidence in the analysis, in the conclusions, also in ultimately in the decisions. So before moving on, in clinical trials, we use the work subgroup analysis to discuss investigations around treatment effect heterogeneity. So in this talk, I will use these 2 terms interchangeably. And there is a very famous paper from Steve Ruberg, one of the big names in pharmaceutical statistics, that having the title Assessing and Communicating Heterogeneity of Treatment Effects is the hardest problem there is. And there is some funny story here. I was talking with one friend of mine, and he told me, Kostas, definitely there are more difficult problems in life than assessing heterogeneity. And I asked him, what do you think it's more difficult? And he told me creating a time machine, which I got double surprised why he thought this thing, the second most difficult problem in life. So lately, whenever humans confuse me, I go to AI, and these seminars have the AI. So probably the only AI bit that I have here is this. So I ask an LLM model, a copilot, what is the more difficult problems in life? And not surprisingly, it didn't give me neither the subgroup discovery, neither the creating a time machine. He gave some difficult mathematical problems that if you solve, probably you will become even a millionaire, or some problems in life that if you solve, probably you will become a happier person, like maintaining relationships. Unfortunately, this presentation will not touch any of these points. We'll switch back to the subgroup discovery. And why it's a difficult problem, it's a combination of two things. In these settings, we have a high chance of false negatives because of the small sample size. The smaller, when you go to explore subgroups, you have a smaller sample size, and you don't have the necessary power to have some strong claims and to have high chances of finding something that it's already in the data. But the most concerning thing is the high chances of false positives because of the multiplicity. So here you can imagine that it's a very big space of possible subgroups that we can explore. And the more you search, the more you find. We can find something that it's maybe even due to the random chance that you have some subpopulation with a very high treatment effect. Now, if you move on and just focus on this population, you enter to this gray area of selective inference, and this can lead to findings that we cannot replicate later. And in the following slides, I will try to keep motivating why this problem is challenging. First, I will show you one illustrative example from clinical trials, from some actual trial that we have. And in this program, it's a phase 3 trial, and we had two identical studies, two identical trials. We call them twin trials with the same inclusion-exclusion criteria. And then in each one, we have 600 patients. So the endpoints, it's rate reduction, the lower the better. And we have also 27 baseline variables that we can use them to define subgroups. And using these 27 variables, we can define 70 possible subgroups. And here it's a histogram of the treatment effects in the subgroups and the overall. So the overall, it's almost 0.78. And you can see that there are some subgroups with a very high treatment effect, like the one to the left, 0.55, and with a low, 1.2. So as you expect, you have subpopulations and you have this distribution. Now, I just focus in the one trial, in the study one. Now, and I plot the same subgroups, the treatment effects. And ideally, you want whatever you see in one study to see it also in the study, which, as we said, it's by design, it's identical. They have the same inclusion-exclusion criteria, but different population. And here I will focus in this dot. This is one subgroup. You can imagine it's the women over 50, it's one variable. It's the women, the subgroup of the women, as an example. And we have this high treatment effect in study 1, 0.60. So we will have expect that in the study 2, for the same population, we'll see some similar treatment effect. And you can see that in the study 2, we have a treatment that it's close to 1. So if we focus only in the study 2, the conclusion is that for this population, the drug is not working. If you see in the study 1, the conclusion is that for the same population, the drug is working. So already this shows you the challenges when you perform subgroup analysis, even within the same safe environment of one study with two trials. And of course, if you move to start defining subgroups with two variables, now in this case, we can have 2,500 subgroups, you can see that there is not so much high correlation between the two studies. And easily you can pick up something that looks promising in one study and you cannot validate it in the other. But while I was talking so far for some clinical trials, this is very well documented in in the literature, also for other types of studies. So back to 91, the authors review 9 papers that they were having some subgroup claims. And for all of these claims, there was some attempt to confirm this claim with another trial. And there was no clear signal in any of them. There was in only 2 out of 9, there was some conditional signal, but there was no evidence in most of them that there is some effect in the subgroups that were reported as promising. And even more recently, Professor Ioannidis and his team, they review 117 subgroup claims out of 64 RCT trials, randomized controlled trials. Out of these 117, 46, they were having statistical significant interaction. So if you just move on with them, you can start making stories that there is some effect in these subgroups. And from these 46, there was only attempts in 5 to replicate the finding through meta-analysis or another RCT, and they failed in all of these 5. So actually, this nicely captures this nervousness that we have when we engage to discussion about subgroups. And the main message is that without external replication or plausibility, just data-based findings can be very, very speculative. Now, I think I spent more than 10 minutes motivating why this problem is difficult, but it's very important also to have some solutions in place for this problem. And it's very important internally, let's say in the company, because a lot of important decisions, they rely in analysis of subgroup analysis. So, for example, we have decisions around enrichment, what populations to target. We have around generating hypotheses for new trials, or we can have also analysis to prepare for interaction with the health authorities. And while it's important internally, it's also very well documented that it's also important externally. And this is some papers and white papers from regulators that they provide some guidance and insight about subgroup analysis. What I like, it's one quote of Rob Hemmings from the last paper that he mentioned that ignoring the problem and similarly dismissing the results of subgroup analysis is not a scientific solution, because there are a lot of people, and there's a lot of people that strongly believe that maybe it's better not to perform this analysis at all, because as we said, it's so challenging. But on the other hand, I think I identify myself with this quote, that it's important to have some solutions in place that you feel that they can provide some insights by accounting to all of these challenges that we discussed so far. And this first part will be about this. So just a bit to summarize, when we focus in subgroup analysis, the data that don't speak from themselves, because we haven't designed the studies to answer the question around heterogeneity. The primary objective of the study is different, to give some sort of, in the clinical trials, to give some evidence of the treatment effect in the population of the study. So we haven't powered towards subgroups most of the times. And as I said, we need to take external information to account. And this can take the shape of many different. It can be historical trials from similar drugs in the same indication or same drugs in other indications, preclinical information, mechanistic information, and of course, biological plausibility. Without this external replication or plausibility, database findings alone can be very speculative. And we need a systematic framework that considers all of these issues. Sorry? Okay. Now, this can be also the case in most of the exploratory projects, that we have the tendency to focus a lot in the analysis part, machine learning, statistics, modeling. But in order to get the most out of this project, we need to approach it more holistically. And we need to have in place some structured framework like the PPDAC. And here it's the great book of David Spiegelhardt that it's about introduction to statistics, that from early enough, he emphasized the need to think it not in terms of analysis, p-values, models, in terms to think it in this structured way. Start with a question, what problem we want to solve, planning, data, analysis, and conclusions and communication. So this was the inspiration behind our workflow, this PPDAC framework. And we believe that having a framework like this or a systematic approach like this will foster transparency, can reduce misunderstanding or errors when we perform the analysis, and ultimately it can lead you to some better replication of the findings. And here's how the WATCH workflow looks like. So it's 4 steps. We have the planning, then we have some initial data analysis, then it's the core analytical part of exploring the treatment effect heterogeneity, and then we have the communication and conclusion through this multidisciplinary assessment. And I will go to these steps one by one, but the ultimate aim of our workflow is to generate insights on how treatment effect varies with baseline characteristics. We want this to be pre-planned, and the results we want to be quickly available after the database lock. It means, after we have collect the data for the clinical trial and we start the analysis. So we want to provide these insights quickly to the teams to start, not to leave, the teams start creating stories from findings that maybe, they're not so trustworthy. And as I said, we want to combine external evidence with database evidence to improve decision making. Our scope is this exploratory assessment to be applicable in phase 2 and 3 trials, and out of scope it's to provide confirmatory statements on subgroup effects. And now let's go step by step. In the analysis planning, we involve and align with the stakeholders and the subject matter expert about what's the question. And in our case, this can be the statistician of the trial, the clinical scientist in the trial, maybe regulatory expert. It can be a diverse group. Most of the times it's a diverse group. And we want to understand what is the outcome variable of interest. If we have more than one studies, which studies to include, in which scale we are interested, what is the treatment effect heterogeneity scale that we are interested in. It can be in odds ratio and the risk difference, depends on the study and the interest of the team. Again, what kind of comparisons we want. If there is more than 2 arms, we want to explore, treated versus placebo. We want to explore subgroups with respect of high versus low dose. And then the more critical thing is to identify which is the baseline variables, the features in machine learning terminology that will include in the analysis. And before running the analysis, we want to see what is the a priori external evidence that the team has that maybe these variables can act as an effect modifiers. And this can be, from previous trials that we have, from the public, from the, or from, as we said, from the mechanistic understanding of the drug and so on. And we want to document all of that before running the analysis. We want to have documented this, all of these aspects, and also in the levels, we want to have some different levels of how these variables can be act as an effect modifier. And then we move to, let's say, to start preparing the data for the analysis, which we call initial data analysis. And here we want to explore the data without touching the question, without touching the question of subgroup analysis. We want to understand how our data look like through producing various visuals, and then take some actions to pre-process the data. And to this end, we follow this initial data analysis framework that it's one nice way of the researchers to interact with the data responsibly before touching the actual questions of interest. So what we are currently provide to the teams, we provide various visualization where they can see, let's say, the distributions, the missingness, when there is sparse categories, or when there is, as I said, missing the correlations, and then they can take some decisions before running the analysis. And the decision can be, transfer some variables that maybe they were skewed, exclude variables that they have very high missingness or very low information, in the sense that if you have a categorical variable that one category is, very rare, if you maybe you should take some actions there, or merge it, or just ignore this variable. When there's highly correlated variables, like the case, the BMI and the weight, we want to understand with the team if they have any preference in one or the other. And of course, when there will be some missing values, even in these clinical trials, a lot of times it's very expensive, and you try to collect all of the information, you'll have some missingness, then you need to take some action to impute the missing variables. And then when it comes to the outcome variable, again, you need to impute it if you're going to following the estimate strategy and all of these things that they do in the primary analysis. So after this step, we have prepared the data set to be ready for the analysis. And here it's the core analytical part. of the assessment of heterogeneity. And we split it in 3 questions. In the first one, we want to perform a global assessment that captures the overall evidence against the null hypothesis of homogeneity. Then we want to identify which are the drivers of heterogeneity, the effect modifiers. And then we want to provide insights how the treatment effect changes with the unidentified variables. And we had performed some benchmarking of methods that were published in the literature across these 3 questions in a previous paper. And this was also the driver of splitting the problem in 3 questions. We felt that each question provides some unique insight to the team. So it's important to have it in this level. And we start with this global test. The first thing is, well, okay, what do you define as a null hypothesis? So in the clinical trials, because they are designed with some specific population in mind, we feel that it makes sense to have the null hypothesis that this population will have some homogeneous effect. Because, you know, if you are expecting something dramatically different, probably they would have adjusted the inclusion-exclusion criteria. So our null hypothesis is the effect is homogeneous. And then we have want to have a global test to test this null hypothesis, which also adjusts for this multiplicity adjusted. So if you put a lot of variables, you know, you don't want to drive the conclusion just because of the chance you search many things and you found something. So we want a global test which is gives you some p-value that is adjusted for this multiplicity. And the other important thing is we don't want to interpret the p-value as a way of taking binary decisions, you know, above or below of a given threshold, 0.05. Because as I said, it's an exploratory analysis. We are not powered for this binary decision making. So we want to see it as in the continuous scale as a measure, let's say, of divergence between the homogeneity assumption. And now we try also to work a bit some ways of interpreting the p-values that maybe will be easier for the teams to digest and provide some sort of levels. And we connected with the notion of surprise, which also it's in information theory. It's well studied, but also this paper from Greenland's group, it was a nice inspiration. So a p-value, for example, of 0.063. What does it tell you? So it will be, you'll get the same surprise if you see this p-value under, let's say, the homogeneity, with the surprise that we'll get if you throw a fair coin 4 times and all of them there lands as a head, or 0.001, it's 10 times. So what do you feel that is more surprising? If you throw a coin 4 times and have head, it's already, you start getting suspicious. Okay, 10 times, you're, you know, maybe it gives you even more confidence that this is not because of the chance, this finding. And we try to provide also some verbal summary of the evidence to the teams to make it easier to communicate. Then after this, first we move to the identify what is the drivers of possible heterogeneity, and having these drivers, they can help us later to define subpopulation of heterogeneous treatment effect. And to this end, we use some machine learning modeling, and I will give more details of some implementation in the second part, that we provide some sort of a ranking according to how strongly the variables modify the treatment effect. And then once we have identified promising effect modifiers, we provide visualizations to the team of how the treatment effect changes with the identified modifiers. And this, as we said, we don't want to use it as any sort of a confirmatory conclusion. We want to have it as a starting point for the team to start discussing and trying to, you know, to get some informed decision from the data. Now, and this comes to the last step of the workflow, which is this multidisciplinary assessment. And we present the outcomes to cross-functional teams that they assess the credibility. Again, we emphasize that the background information that we have documented in the planning stage, it should play a critical role when you assess the findings. And as I said, we want to avoid confirmatory language, and we want to emphasize the interpretation in the context of what we had documented in the beginning. And overall, we can say that the main purpose of GOTS is to interrogate the data, generate insights, and of course, what it comes next depends on the insights and the specific situation of the program and of what the team is interested. So here is some possibilities that we have in practice. A lot of times, this is provides some insight and we see some unexpected findings. So we repeat some analysis to explain this finding or to try to provide some insight in this finding. Then we have this, the team comes up with new endpoints or new variables that they want to explore. A lot of times they are happy with the initial analysis and nothing comes back, comes afterwards. And sometimes, you know, if they want to quantify the subgroup treatment effect, then we need to use some ways of, we have some methodologies to provide some adjusted estimates of the efficacy in the specific populations. So what I want to say also, now this, if you remember when we have this PPDC is defined also as a cycle. You know, you have this planning, data analysis, communication, and of course, in the exploratory projects, of course, we can end up in this cycle. So most of the times in practice, you have this cycle, which is motivated again with some other question of interest from the team, and you keep working on that. So this is a bit of the summary of the first part. We believe that GOTS provides a systematic approach to explore treatment heterogeneity, and you can nicely combine external evidence with a data-driven insights. And it's we have put it in place with various versions. This is the last version that I present so far, but we work with these questions, let's say the last 2 years at least. And we have engaged in various projects in phase 2 and phase 3 across disease areas. And we try to provide insights around heterogeneity, around arms, which arm may respond better. Another important application is to identify populations that manifest specific adverse events. This can help you, for example, to position one drug in some subpopulation without the need of worrying for post-dose monitoring, for example. This can be another application that it doesn't have to do with efficacy, but with the safety. And also it is very helpful to explain regional differences in these multi-regional trials. So these are global trials. A lot of times you see in some specific countries some results that they are not in line with the overall results, and then maybe you can use this information from the effect modifiers to dig deeper and to see if there is some difference in the distribution of the effect modifiers that can explain why in this specific country we see something different. And this is some example applications of the workflow so far. So now I will move to a bit more of the technical one, implementation of the workflow, that it builds upon the causal inference. But do you want me to pause a bit if you have any questions, or we can take them at the end, whatever you feel like?
Quick question for you. How long is the second talk?
It's around 20 minutes, I'd say.
Well, probably best to do because we've got to vacate the room at 12.
Okay.
Yeah.
Okay, perfect.
What time the questions at the end, we'll do that way.
Cool. Nice. Yeah, the second part actually is the first time that I present, so I'm not sure how well I have calibrated. So let's give it a bit of time. And yeah, here it's the GOTS workflow. And now I will focus in these 3 questions, which is actually the, let's say, the analytical part of the workflow. And we'll focus on building on the idea of inferring some individual treatment effect. And now I have some visual, let's say we have this population in our clinical trial. In the high level, what we do, we split it at random to half. In this case, the left group takes the treatment, the red drug, and the right one, let's say, takes the placebo, the blue drug. And the approval, it comes in the primary analysis in the overall population, can be based on something like this, the average treatment effect. We can see this as the expectation of the Y, the outcome, when you take the drug, minus the outcome when you are in the placebo. But while this is, when you see this makes sense, the first time that I saw this image, say, okay, maybe I'm interested more in seeing what is the treatment effect for this person with this characteristics. In this case, we have the same hairline. I don't know why. I said, okay, I want to see the expectation of the drug, how it will improve in this person. And this is what we call the conditional average treatment effect. So this is the expectation of the drug, of the efficacy, conditioning to some characteristics. And this moves from this overall understanding of how the drug works to more individualized understanding of how the drugs work. And this is the driving force for us to answer these questions. Now, a bit of a notation. This X is the baseline covariates, the features, and Y is the target, the one that we want to see how it's one, let's say, captures the disease severity. And the X's that they're highly correlated with Y, we call them prognostic variables. And this is the traditional, let's say, machine learning data set with X and Y. And when we perform feature selection, we want to identify which X's are correlated with a Y. Now, in the clinical trial, we have an extra variable, which we use. It's the intervention, the treatment, A in this case. And half of the people take the red pill, and the other half take the blue pill. And there are two outcome variables which it's not fully observed. The Y1 is what would have been the outcome if the person was in the treated group, the red pill, and this we observe it only for the half of the population, and the Y0, what would have been the outcome if the person was in the placebo. And ultimately, in the personalized, let's say, scenarios, we want to identify what would have been the treatment effect for each specific individual. And this is the Y1 minus Y0, which we don't observe. And this we call the fundamental problem of causal inference, that you see one part of the reality. We don't see both. But this is, for us, this is of interest. And on top of that, we want to identify also which is the baseline variables that they are influence this. They are, let's say, correlate with this treatment effect. And with this, we call them predictive variables of effect modifiers. And this is what we try to identify, let's say, through the WODs in the second part. Now, there's various ways of estimating the treatment effect. I will not go into details, but what I will discuss about three of them. The first one is it's also called G-commutation or outcome modeling. You build two models, one using the data from one treatment, the other from the other treatment, and then you make, let's say, the prediction for each individual, and you sum across all of the individuals. A different way of estimating the average treatment effect is to build the propensity model, which is actually the model of predicting the treatment given the X's. And using this model, you re-weight the outcomes, and then again, you can estimate the average treatment effect by summing all of the across the patients. I need to, sorry, I forgot to mention that these models, this way it's consistent if you have a well-specified the outcome model. This is the propensity score model, and there is a combination of these two, which is called also augmented inverse probability weight, which is you can see it as the inverse probability weight corrected for something that reminds a bit it builds upon the outcome modeling. And this is quite a robust way of estimating the average treatment effect. It's also double robust estimator because it's ensure consistency, even if one of these two models is not well specified, it's misspecified. And at the same time, if all of the models are well specified, it's it has faster, improves the efficiency, which means that it's, in simple terms, it has smaller error. So this is one way of estimating, three ways of estimating the average treatment effect. Building upon these ways of estimating the average treatment effect, have been suggesting some ways for estimating this conditional average treatment effect. And in this talk, I will focus on this double robust, which essentially is the summand of this AIPW, what we see inside here, and it's called it's called pseudo outcome. So you have something that you can be seen as a proxy of the individual treatment effect, and then the idea is you have this and you regress over it in order to derive the Kate. And here, in all of these boxes that I have in different color here, it's a different, let's say, machine learning models. It's a traditional machine learning model, regression model. So in order to estimate this Kate, essentially you need to build 4 different machine learning models. And in a very, a nice paper, Kennedy have suggested some ways of estimating this double robust estimation for the Kate, and this is the algorithm. So what I will try to present in the high level, what we are doing, we split the data in, let's say, in train and test, or we have this faults, like in the cross validation when we want to tune hyperparameters. We use the training data to train the nuisance functions, the propensity score and the outcome model in the treating and the placebo. And then using this, we estimate the pseudo outcomes. And then in the test data, we train the last model, the Kate model. So, and then we have for the test data, the predictions, and we repeat this process across all of the faults. So Kennedy shows that having this type of cross-fitting reduces the chance of overfitting and improves the quality of the estimators. But the main outcome of this procedure is that you have, for each patient, you have a pseudo outcome, Y, but also you can have also the Kate, the estimation of the Kate. So as I said, all of that can be seen as a different machine learning model. We have 4 machine learning models that we need to train. And in our implementation, in our work, we use this idea of the stacking of the machine learning model, the super learner. And this process combines different models and then reweights them in the prediction from the different models. They are getting weights that they are, in some clever way to minimize some cross-validation error. You can be, you can go, you can be aggressive, you can fit many models there, you can be more selective. In our works, we use 2 different types of modeling. One that it builds upon the linear modeling and they have this regularization, the lasso, and the other one, it's one version of random forest. But as I said, it wasn't our optimal goal to find what is the best type of model, but we try to find also some trade-off between simplicity and also having some nice performance. And so combining all of these ingredients, now for each patient, we have a proxy, let's say, of the individualized treatment effect, which is captured by this pseudo outcome, which is one of the outcomes of this double robust learner. And then we want to use this as a driver to answer these 3 questions. Firstly, we want to use this to perform some test of independence between a multivariate space X, the space of the baseline covariates, and this new outcome that we have, the psi, the pseudo outcome. And we define some desirable properties for the p-value. Firstly, when there is no heterogeneity in the data, we want this p-value to be uniform. And when there is some heterogeneity, we want the smaller the p-value, the better. So to test this multivariate, let's say, hypothesis, we use some, maybe I should have emphasized all of this code, it is based in R. We use the package of coin that is from Hawthorne and his group, and essentially it's a permutation test. But it gives a lot of also variations. So we focus in 2 variations of this permutation test. One is the one statistic that is driven, let's say, from some extreme values in the covariate. For each covariate, you calculate the test statistic, but it takes from all of this statistic the maximum. So if you have a scenario that you feel that is a single driver of heterogeneity, the maximum type of statistic, it will be more powerful. Now, you can have some scenarios that you feel that collectively a lot of variables contribute to heterogeneity. This is another statistic, it's the quadratic. And we ran some simulations. I will not give all of the details of the simulation scenarios, but the simulation scenarios, we published in the previous paper, they are motivated by the clinical trial. So the X's come from a clinical trial. And then we build some functional form towards the Y that we can control. So in this case, this is when we don't have heterogeneity, the treatment does not interact with X's. And we can see that both of these tests, the distribution of the p-values across this 500 simulation, it's uniform. So the tick our first objective. And then when there is heterogeneity here in the x-axis, we start moving towards scenarios that from 0, no heterogeneity, to 2, a strong heterogeneity. And then we see the lower the better. And we see that the maximum statistic outperforms the quadratic. And this has to do because in this scenario, there is only one driver of heterogeneity, the x14. So now if you have many variables and functional form here towards that they interact with a treatment, maybe the quadratic statistic will perform better. But in the paper we discussed 4 different scenarios, and overall, at least in the settings that we focus, the maximum statistic was the preferred one. Now we have one way of answering the first question, giving a p-value of this global test for heterogeneity. Then we move to identify the drivers. What is the effect modifiers? Again, here we approach it as a machine learning problem. We have the X and the Y, and we want to derive some important scores that rank the X's according to how correlated, let's say, they are with the Y. The property that we want this time is to be unbiased, because in the clinical trials, we have a mixed type of data set, continuous and categorical. We don't want to have some methods that is favor one against the other. So we know there's well documented that the off-the-shelf random forest importance scores, for example, they're in favor of a continuous variable because it gives more splits and can lead more purity. We don't want something like that to happen. So we checked 2 different ways of deriving importance score. One was using this conditional inference trees, which is a version of random forest that before the splitting, it performs some tests, and then according to this p-value does the splitting, and actually it's more robust towards this bias. And then we use this permutation importance. And then the other way, it was using the random forest, the traditional random forest, and then on top of that, having some way of deriving importance scores, again, motivated by game theory, but again, widely used in machine learning. So, and we have 2 scenarios again. When we don't have heterogeneity, we want the methods at random to pick the variables in the top of the list, because there is no heterogeneity. So every time that we run, maybe a different variable will pop up in the top of the list, but we don't want to have some systematic biases there. So we can see that the random forest, the circles are the categorical variables in the data set. They tend to select continuous, the crosses, than categorical. While, as I said, in this case, we want everything to be in this dashed line. And then, when we have heterogeneity, again, there is some scenarios here. We explore that there is in the predictive part, there are 2 variables, one categorical, one continuous threshold, and we saw that this idea of using the permutation score around the C-forest outperforms the other. But as I said, it's not universal. In the paper, you can find scenarios that also the sub-values around random forest. There is some scenarios that they perform better than the permutation C-forest. And finally, we want to see how the different methods, let's say the quality of the K estimates. And here we compare the DR learner with the components, which is the IPW and the T learner, which the T learner actually in the clinical trials literature is also known as the virtual twins. And again, you can see that the DR learner is having a smaller error. And we see various scenarios without heterogeneity, with a strong heterogeneity. And then around cross-fitting also, it's another degree of freedom, and there are papers that they suggest different ways of cross-fitting. In this work, we compare a couple of methods, and we pick up the one that was performing better. So overall, we had now one way of using this DR learner to answer the 3 questions of interest in this SWOT framework. And then we compare with other approaches in the literature. As I said, this is also such a challenging problem. You shouldn't expect that there is some silver bullet. There is all of the methods. There are scenarios that perform equally well, but overall the DR learner have some decent performance, and also it was ticking some other boxes that was important for us around computational complexity, and it's quite adaptive when we use mixed trials, and it's adjusted for confounding and when we use it in observational studies. So this is the preferred approach, and we put it in practice in some clinical trial that we have in psoriatic arthritis. psoriatic arthritis is an inflammatory disease and affects severely the quality of life of the patients. Novartis has a treatment, Cosentyx, which is approved for adult patients with active psoriatic arthritis. We had 5 phase 3 trials that we have a big sample size. It's around 2,000 patients. The endpoint that we focus is one composite endpoint that takes a lot of things, and then it's a binary 1 if the person responded good, 0 not good in the week 16. So the Y it's binary essentially. And then the estimate that we focused was this risk different. What is the probability of responding if you are in the drug minus the probability of responding if you are in the placebo? And we have 70 baseline covariates to explore. And this was mixed of categorical and continuous. There was demographic, lab values, clinical measurements, and there was also one proteomic marker. And there are 3 works published from Novartis colleagues in this data that they were already focusing the question of predictive biomarkers. Essentially, one of the works that we had was using some machine learning methodology that controls the type 1 error known as knockoffs. And this was, let's say, the promising, the variables that this in this paper they were identified as effect modifier. Then there was another paper which was actually quite interesting. Internally, we ran a big subgroup challenge that many teams participate across this part of the organization, and here they provide their solution of what they think it's a subgroup of enhanced treatment effect. And here it's some sort of a ranking how often they use these variables to define their subgroup. So the first 2, it's the same. It's this reactive protein nates, then this is the fatigue score that lands here, and so on. And then there was another work from a different group in the organization that they focus in proteomic markers for this trial and identify maybe proteomic markers that they can ask as an effect modifiers. And they found one promising was the BD2. So now we have these 3 works. And what we do essentially is we want to use the WOTS with a DR learner to see if we find similar hits with that. First of all, we apply and we have a very small p-value from the overall assessment from our first objective, which is reassuring because we knew that this is quite there was heterogeneous treatment effect. Of course, the good thing is that the heterogeneity was quantitative, not qualitative. Always in this population, the drug was helping the patient, but the extent of the treatment effect was changing. Then this is the ranking that we derive using this DR learner, and on top of that, the permutation and the conditional random forest. We see that the first 2 hits is exactly the 2 hits that we had in the previous work, the C-reactive protein and the age. The third one is the proteomic marker was reported in the third work. So overall, we see that this method returns something that it's sensible and something that we were expecting. And then, of course, we want to see how the treatment effect changes with identified variables. And here we have 3 groups of the people with a C-reactive protein. We see that the higher the inflammation essentially in the baseline, the more the treatment effect. And with a star also, we have some estimate that they come from the K, which you can see them as an adjusted estimates of the treatment effect. And we provide something similar with for the proteomic marker. So just now a summary. Understanding how treatment effect varies across patients, it's a very important question and influence a lot of sponsored decisions. We have this workflow into place that will provide some systematic approach to combine external evidence and the best scientific understanding to solve this problem. And then we saw in the second part 1 implementation of the WOTS workflow, the analytical part, building up on the double robust learner idea. And I want to emphasize this is a joint work. We are a team of 5 people that we are based in this methodology group, and we provide support to our trial statisticians. And thank you very much.
Thank you very much for that, Kostas. We've got 4 minutes. Does anybody have a quick question? Yes, we've got a question at the back.
Yes, I have a question because here, as we are aiming to estimate the causal impact of the treatment, so here we apply AI algorithm sort of to identify the important.
Hang on one sec, Kostas, can you hear that?
Not very clear. I hear you more clear.
Just want to look because it might be easier. Yeah. Oh, that's it. Put it on the spot.
Oh, I can hear me now.
Yeah, now I hear you better. Yeah, thanks.
Okay. So I have a question because here mostly about the second part to estimate the treatment effect. So it is under a causal framework, and however, in the clinical context, we are more interested in because you apply propensity matching after you do the AI selection of the important covariate, but we are interested in how this covariate change or the causal structure behind underlying the mechanism of the disease. So I would say, how would you like to explain, make it more expandable, make people to understand how the treatment, because it's not like in data imaging, like where the AI is normally working on. So you just analyze the image and using algorithm to see. But here in the clinical trial context, slightly different. I think we are more interesting and want to know how mechanism interaction with each other and that cause the change in the treatment. I don't know. I think it's not like a really clear question, but this is my.
No, thanks for the question. Now, it's true that we don't assume any, let's say, functional form. We try to do a bit of more non-parametric, but if you want to see how the variables interact with the treatment. Another thing that I haven't mentioned, that sometimes we use also this interaction approach, but to see if there is pairwise interaction towards the outcome. But we don't have something in place for that specific question. No, sorry.
Like there is a large amount of misses in medical data as well. So what we are doing now is just analyze for covariate. Like when we do the analysis of CVD, cardiovascular disease, only BMI, cholesterol, and blood pressure, and we try to analyze whether there is like a collider bias, like just trying to underline what's their relation. So it's
but in our studies in the RCT trials, you don't have the collider in the sense that you have the baseline and the outcome, and there is no, you don't have. Now if you want to see, probably you discuss in the cardiovascular disease post baseline how the drug helps the CRP and how the CRP then influence the outcome. This is not touched from this workflow. We don't include post treatment variables. All of them, it's before the person starting the treatment. I can share with you some publication from a colleague in cardiovascular that they were trying to define subgroups. It has to do with this principal stratum idea. So there you can say something under some very strong assumptions, and then always this thing, maybe it's criticized because how these assumptions can be plausible. But sorry, I didn't mention. I thought it was clear that we don't talk about post-treatment variables. Everything is before the person starts the treatment. So we don't want to see how the treatment through which pathways affects the outcome. We have the baseline variables and the why. So we don't include in the analysis post-baseline.
Thank you.
Thank you for your question. We're going to have to wrap it up now, unfortunately, because we've got to hand the room over to somebody else at 12.
Okay, sorry, didn't realize.
No, that wasn't you at all. You did the talk was fine. Just one more time, just everyone say thanks to Kostas for joining us today. Really appreciate it. I'll stop recording. Actually, I should say, I'll be sending the recording out via the usual various channels. So
and if you have any questions, feedback, feel free to reach out in my email. You can find it through John or in the webpage. It's everywhere. Okay, thank you very much.
Oh.